what's an arms race, and what aspect of this system seems like an arms race? Trevor, want to start us?
An arms race is when like two opposing forces are constantly trying to like out the other person with power. And so in this case, the newts are trying to get more and more poisonous, but then, or hypothetically, the newts are trying to get more and more poisonous, but the snakes are trying to get more and more resistance. So they're kind of like always in an even place.
Yeah. So it's easy to talk about it that way, but I want you guys to get familiar with or recall that the key here is reciprocal selection. So there's no trying. This is a consequence of variation and differential fitness. I'm only picking on Trevor because he had a really good answer, except had that word try. And it's tempting to emphasize these things, but know that it's a process that happens at the population level. It has nothing to do with the preferences of the individuals. Yeah. So we've got reciprocal. So can you guys think of any prey that that adapt to predators, and you can see evidence of their adaptations, but you're not seeing an arms race? Yeah.
The lizards and the loggerhead sharks.
Oh yeah, that's a great one. Okay. So the horned lizards with their long horns. When you have that kind of evolution of directional selection towards more and more extreme prey defenses at some point in the predator-prey sequence, we call that escalation. So directional natural selection can lead to adaptations that can give you extreme phenotypes, whether that's really long horns or really toxic newts. But when it's just a one-sided thing, we tend to call it escalation. And they think they talked in the beginning here about observations of escalation in marine organisms during the Cambrian explosion, when predators evolved eyes that were better able to focus on and detect prey. We see escalation in prey evolving to avoid visual predators. But it's hard to know if that's actually an arms race, if the prey being able to better adapt to a visual predator means that predators becoming more sophisticated. That might be what happened, but it's difficult to test in the fossil record. So here we have a chance to look for reciprocal selection on both sides of the system and determine whether, in fact, we see an arms race. So, reciprocal selection. And what about the number 3? Why this newt? Why Ptericha granulosa? It's not the only newt. It's not the only organism that produces TTX. Why is this a good one? Why this system?
Isn't it one of the only ones that has a predator?
Yeah. Yep. You need to have 2 players to have the arms race. And we knew from Brody's earlier work, Butch's work, that these snakes are eating these newts. Locomotor performance. If you need to know how this toxin affects the snakes, why locomotor performance is a good choice? It's because it's a neurotoxin. Yeah. So we've got a neurotoxin. It's going to affect the muscle performance and motor performance. Okay, and why did they use young garter snakes? So their experimental design, they didn't just go out and get snakes randomly and supply apply the PTX, let's talk briefly about the design. There's two kind of groups that they use, two kind of groupings that they use. What are the two levels of grouping that they use for this experiment? What's the experimental design? I'll turn that back on in a moment. Right. There's geographic groups. What were those geographic groups?
Oh. The Idaho.
Who's in Idaho?
Idaho, the T-shirt towels.
Yeah. And then over here in Oregon.
T-shirt towels and T-Oregon.
And so we got three groups of snakes, but within those three geographic groups, there was an additional grouping variable. What's the other grouping variable? This is the sympatric-allopatry. So if our newts are just along the coast here, we've got two groups that are sympatric and one group that's allopatric, one group of snakes that are sympatric allopatric. But within these three, there's another grouping variable,
The families within.
families. If you think back to natural selection, what other thing do you get to learn about by using families?
Heritability.
Yeah. So how would you expect heritability to express in, say, the sympatric T. sertalis? How would you expect that to see that in these different families? Or what's another way to say that? How does having siblings allow you to detect heritability? Siblings in the experiment.
You could compare it to like the mother and see, how much of that trait did the babies get compared to the mother or the siblings.
On average, what's the genetic similarity between a mother and offspring?
Half.
About half. On average, what's the genetic similarity between two full siblings? They're each going to get half their genetic information from a parent,
So probably like a
but
fourth.
which half is random? Which of the chromosomes they get, right? Because of that independent assortment of the chromosomes. It's gonna be more like half. Right? Because if you're identical twins, then you got all the same, right? But because which chromosomes you get is random, because the tetrads randomly assort during meiosis, you can actually just compare the siblings to each other. You don't have to compare them to their mother. And you would expect siblings to be more similar to each other than they are to other members of other families in the same population if there's heritability. Did they find a significant effect of family when they did ANOVA on resistance? The graph is nice, but reading those ANOVA tables is a little intimidating. But you guys have learned ANOVA tables, so you can do it. So you can see, is there a significant effect of family in table 2?
I thought there was, but I didn't read that one in the Nova
Is basically I'm asking, is the litter effect significant? Yeah, so they have this rough estimate of heritability, but what I want you guys to understand is just that there's an effect of litter on resistance. It's not just whether you eat newts or not. There's also variation among families, which suggests that there's heritability. So, hold this back up. have one question once we hit this. So here Paul's writing down the ANOVA. And I have one more question for you all after we make sure this is clear. So are you looking at table 2? Is there a significant effect of litter? Yeah, it should be on the bottom. Right, so effect of litter height, so snakes from the same litter are more similar in their resistance than they are to, than you expect by chance if there was no effect of litter.
So the way this works in an ANOVA, remember that the ANOVA calculates how much variation there is among litters versus how much variation is unexplained or is within litters. And that gives you the total amount of variation. If there's a lot of variation that is accounted for by this division of the individual snakes into litters, then this is going to be big, this is going to be relatively small, this is going to be big, this is going to be small, this is big, this is small, and we see these large F statistics. So whenever you see a lot of the variation accounted for by the fact that this snake is a part of this litter versus being a part of this litter, that's telling you that the family component, which is gained by either having a common genetic background or having a common environmental background or a combination of both of those, that's a large effect, and the random variation is relatively small. And so family members resemble one another because of common genetics or common environment or both, and that comes out in the ANOVA in the litter effect. And that's one of many ways of determining heritability.
Can you guys think about, as we get ready for the rest of the papers we're going to read, what sorts of things might bring a predator-prey arms race like this to an end? We've got the snakes becoming more resistant. And, well, I guess I was going to ask about the newts too, but I'm worried about time. So I talked with each of you, what's the logical next step you want to know about if you want to investigate this arms race? Did they show variation in heritability in the snakes? Variation in resistance and heritability for the snakes?
Yeah.
Yeah.
What didn't they look into?
Variation versus heritability for the newts.
And if we find that that is the case, as we might expect it to be for a prey that is killed if it's eaten by a predator, how might a predator-prey arms race end? What kinds of things could cause this system to stop being a predator-prey arms race? Yeah.
Maybe the newt could become so toxic that it kills any snake that tries to eat it.
Right, that's one way out. What's another way out?
The snake becomes so resistant that the newts can't evolve any further.
So we're going to see consequences, or we're going to start thinking about consequences of what would happen evolutionarily if one of those things happens. And so you guys have another paper. You have Brody Berry 1999, excuse me, for next week.
No.
And we're not doing a literature summary, but read it and think about it and be prepared to discuss it in class. And we post the questions that we're going to talk about on Moodle, so you can go look ahead and see what those are going to be like.
Monday, Wednesday, and Friday, it's a paper a day. No written summaries, but you need to come in prepared to discuss it like you did today. So even though you haven't written a summary on it, we still expect you to be capable of talking about it. I need your homeworks, and I need your summaries.
I do. Yeah. I only got a photocopy.
When/how can I get my email?
I'll get you my stuff.
When?
Kate.
Thank you.
Sonia?
Do you leave? At Pacific time?
I'll be
that.
coming pretty soon, you're just drop it by.
Yeah.
Okay, I don't have it.
No, I just had questions. So my research proposal thing, do you want me to email it to you or print it out and
If
give
you
it
can put
to you?
it on Basecamp.
Okay, that'll work. And then I had one, so I ran into a problem with our studio again. And it was, so I did the coordinates, I fixed all that, so it's X and Y and Y. But then the problem now is that it won't, it says it
Less of the
won't read it
variation
because it's non-numerical.
than just random variation.
Um,
So,
well, it's okay if you don't have that figured.
so
we'll
That's what I
see.
figured,
So
but
remember
I
that.
just wanted to let you know that, like.
What an analysis.
Another
is
issue.
Is it's looking at, let's see if this is.
But, yeah, and I try to figure it out, I really don't know, so, I'll just figure that's a
Yeah,
problem for
we'll
this
do
week.
it together.
Are
We can get it
resistant,
together.
Some litters
Okay,
are
that's good,
resistant,
and
some
I will
litters
get that
might be
posted
moderately
sometime
resistant.
tonight.
Okay, sounds good.
In all of these litters, we calculate what the grand mean is, and this figure up here is looking at how far are each of these means away from the grand mean. And then it compares that to how much variation is there within means. And the more variation there is between means and the grand mean, this gets bigger and bigger and bigger, which means that these distributions for litter 1, litter 2, litter 3 are further apart from one another. So more of the total variation in the data set can be described by variation due to what litter you happen to come from. And so more of the variation, if it's explained by litter, means that more of the variation is actually determined by genetic variation. Less variation determined by litter means that there's more random variation, which means that there's less genetic control. Thank you. Sure. Did it seem like everybody had a pretty good idea of things today?
I think so.
Yeah, it seemed that way to me.